Welcome back to the deep dive. Okay, today we're heading into the Western Pacific, focusing right in on the Coral Triangle. People call it the Amazon of the Seas.
Mhm.
And there's this really groundbreaking effort happening in the Philippines. They're setting up Southeast Asia's very first coral larva cryobank. We've got some fascinating material here looking at why this region is just so critical, and also the pretty intense science they're using, basically freezing genetic blueprints.
That's right. So our mission today is to really get across why this specific area matters so much globally, why it's such a conservation priority. And then, yeah, dive into the mechanics of how this cryobanking actually works, freezing this genetic material. It's complex stuff. The Coral Triangle itself, I mean, the scale is just immense. We're talking 5.7 million square kilometers. And it stretches across 6 countries: Indonesia, Malaysia, Papua New Guinea, the Philippines, Solomon Islands, Timor-Leste. But the biodiversity, that's where it really hits you.
Absolutely. I mean, think about this. 75%, three-quarters of all the world's coral species are found right there. That's staggering scientifically, yeah, but the human side is just as vital. You've got, what, over 120 million people whose food security and livelihoods depend directly on these reefs. So when we talk about potential reef collapse here, it's not just environmental, it's a massive economic and, well, humanitarian issue, too. Okay, so let's look at the tech side. This initiative led by the University of the Philippines Marine Science Institute sounds like they're using some serious genomics in a race against time.
Oh, they are. They're essentially freezing coral larvae. Think of them as microscopic seeds for future reefs at this incredibly low temperature, minus 196 degrees Celsius. The technique itself is called vitrification. And what that basically means is instead of the water inside the cells turning into damaging ice crystals when it freezes, they flash freeze it so fast that the water becomes like a glass, a non-crystalline solid. It avoids that internal damage.
Got it. So preventing ice damage is the key, preserving the genetic material perfectly. And the idea is these preserved larvae can eventually be thawed out, maybe years down the line, and used to, well, actively rebuild damaged reefs or provide genetic diversity for research.
Exactly that. Both applications are crucial. And it's important to see this isn't just one lab working in isolation. This cryobank is part of a bigger regional network. They're collaborating with institutions in Taiwan, Indonesia, Malaysia, Thailand. That kind of collaboration is absolutely necessary because, honestly, the science here is really, really challenging.
Right, because while you mentioned these reefs took millions of years to form naturally, we're freezing these tiny life forms, doesn't it feel a bit like, I don't know, a high-tech insurance policy rather than a full-scale restoration solution?
Hmm.
Can this cryobanking really match the scale of the crisis?
That's a really fair point. And yeah, the uncomfortable truth is this is primarily about preserving genetic diversity for future use. It's not a magic bullet to fix everything right now. And the science, like I said, it's tough. One of the biggest hurdles is actually the coral's own biology. Many species are just very sensitive to cold temperatures. But maybe even more critically, their cells are extremely rich in lipids, in fats.
Okay, lipids. Why does being fatty make freezing so much harder?
Well, because the chemicals they use to protect the cells during freeze are the cryoprotectants. Think of them like cellular antifreeze. They really struggle to get inside cells that are packed with fat. The lipids sort of repel these protective chemicals. So it's hard to get the cryoprotectant to where it needs to be, protecting the crucial genetic material deep inside the cell.
Huh. That's a serious technical roadblock. And then you've got the whole algae thing, right? The symbionts living inside the coral tissue.
Precisely. Those algae, the symbionts, they add another layer of complication. They also make it harder for the cryoprotectants to work effectively. That's why, as our sources show, the strategy right now is to prioritize certain model species, first things like Bacillopora and Acropora. The idea is to really nail down the protocols on these slightly easier species before tackling the more sensitive, often endangered ones. Oh, and a really crucial detail here. For the whole thing to work long-term, for roof survival, they also need to cryobank the algae symbionts separately. Because if you restore the coral structure but don't have the right algae to live with it, the whole system collapses eventually. You need both.
Okay, so we've covered two big things today. First, just the immense global importance of the Coral Triangle's biodiversity. It's irreplaceable. And second, the really sophisticated cutting-edge science needed just to keep its genetic legacy viable through this cryobanking.
Yeah, and if you step back and think about the bigger picture, there's a real paradox here, isn't there? Nature took millions, literal millions of years to build these incredibly complex ecosystems. And now we're using this ultra-cold technology, freezing life at nearly 200 degrees below 0 Celsius, basically to try and buy ourselves maybe a few decades. The knowledge itself, the fact we can do this, it really throws into sharp relief the contrast between nature's timescale and the speed of the threats these reefs are facing now. Some things, once they're gone, well, technology might not be fast enough to bring them back.